Hello and welcome to the stem cell passaging video tutorial. Passaging, also known as subculturing or splitting, is used to expand cells. For stem cell cultures, it is a useful technique to clean up and remove differentiation. This is accomplished by transferring the undifferentiated cells to the new container and leaving behind cells that have started to differentiate. In order to keep the cells in the log phase of growth, passaging is done before cultures reach 100% confluency. Today we will be showing you two techniques of passaging stem cells, picking and scraping. For the picking method, we will pick and transfer one stem cell colony at a time. This technique is especially useful when trying to clean and isolate a colony from differentiation. For the scraping method, we will scrape all the stem cells at once and passage them together. This technique should be used on stem cell cultures with no differentiation. The stem cells we are splitting today are grown on irradiated mouse embryonic fibroblasts, also known as MEFs. As shown here, properly plated MEFs should be at 40% confluency. The first step in our subculturing is to plate irradiated MEFs 1 to 2 days before the day of splitting. On the day of passaging, replace the media in the MEF plates with stem cell media. Set this plate aside for later use. To passage adherent cultures, the cells must first be detached from the original vessel. In the picking method, we will be using a needle to cut the colonies into smaller pieces. These pieces are then transferred using a P100 micropipette. Gather the required materials. This technique is ideal when passaging fewer number of colonies or when the extent of differentiation is significant. Start by removing the needle from the wrapper. We will be using a syringe as a handle to make the needle easier to hold. Insert the needle into the syringe. Hold the syringe as you would hold a pen. Center a colony you want to passage in the microscope's view. Using a steady motion, draw parallel lines through the colony. Here are a couple images illustrating the parallel lines. Next, rotate the plate 90 degrees. Recenter the colony in the microscope's view. Once again, draw parallel lines through the colony to form a grid-like pattern. Here's a properly constructed grid. The lines of the grid should be 1/100th of a millimeter apart. Each piece in the grid should contain approximately 100 cells. Using a P100 micropipette, nudge the pieces and transfer them into the previously prepared MEF plate containing stem cell media. Generally, pieces from 3 to 5 medium-sized stem cell colonies can be transferred to one wall. Once transferred, shake the plate back and forth side to side to ensure that the pieces are not clustered together. Label the plate with the cell type, the passage number, the date, and your initials. Here's what the pieces should look like in the new MEF plate. Leave the plate undisturbed for 2 days in a 37-degree incubator. Here's the piece 2 days after picking. In the scraping method, a scraper is used to detach the cells and a serological pipette is used to mechanically break apart the colonies before replating. Gather the required materials. The scraping method is used to passage large number of cells with no differentiation. This is what a stem cell culture that is ready for scraping looks like. Notice that the cell culture is at 80 to 90% confluency. Remember to spray all objects entering the hood with 70% ethanol. Start by holding the scraper in one hand and grasping the plate in the other. Press the scraper firmly against the well and push it across the surface to detach the cells. Rotate the plate while scraping to ensure that the entire well is scraped. After scraping, the layer of detached cells should be visible in the well. Next, use a serological pipette to transfer the contents of the scraped well to a 50 ml conical tube. Allow the vial to sit undisturbed for 2 to 3 minutes so the cells can settle. When the cells have gathered at the bottom, aspirate the media from the vial. Using a 10 ml pipette, add 2 ml of fresh stem cell media and pipe it up and down 4 times. The automatic pipetter should be set at medium-medium setting as shown here. This process breaks apart the colonies for replating and resuspends the cells. Transfer the cell suspension to the previously prepared MEF plate containing W8 media. Depending on the initial confluency of the stem cell culture, this cell suspension could be divided equally between 2 to 3 wells. Rock the plate back and forth and side to side to redistribute the cells evenly across the wells. Remember to label your plate. This is how the cells look under 5x magnification after replating. Here we see the cells 2 days after scraping.